Good morning, students. I am LOKNATH GHOSH from Bharat Academy Sciences. I am a biology teacher here. Today I am going to discuss about the photosynthesis. Okay. Now students, you know about the photosynthesis process. This is a very important process in our ecosystem. You know there are multiple factors present in the ecosystem, like our ecosystem starts with a producer and it goes up to the top consumer. And what is the importance of producer? They actually produce energy for the rest of the trophic levels. So green plants are called producer. Why they are called producer? Because green plants are the only living organism in our food chain that have capability to produce the energy by the process of photosynthesis. Now why they have such capability? Because they have chlorophyll within them. So our today's topic discussion is the importance of photosynthesis and the important factors of photosynthesis. The first factor is definitely the chlorophyll. The second factor is the sunlight, and the third most important factor is carbon dioxide. So one by one we have to prove the importance of these three factors in the photosynthesis. Now come to the first slide. Experiment number 1, that is chlorophyll is necessary for the photosynthesis process. What is the aim of this experiment? To demonstrate that chlorophyll is essential for the photosynthesis. Now we use a variegated leaf. What is variegated leaf? A special kind of leaf that have some portion green color and some portion have pale yellow color. Now you know all that the green portions, why the leaves are green in color? Because they have chlorophyll. Due to presence of chlorophyll, they appeared as a green color. So the yellow portion or the pale yellow portion, they lack the chlorophyll. So we have to prove it by experimentally. Because right now we are discussing about, yes, the yellow portion is lack of chlorophyll, the green portion is not presence of chlorophyll. So how we can prove it? This experiment. So in this experiment, take a potted plant with the variegated leaves, what I have discussed right now. D-starch. What is the meaning of D-starch? D-starch means to remove the previously formed starch, because they are living matter, they are living organism. They continuously what can I say? They continuously photosynthesis is performed within them. So if photosynthesis is performed, that means C6H12O6, that is the formation of glucose, is continuously there. So we have to erase out the previously formed starch or previously formed glucose. How? That process is called D-starch. D-starch by keeping in the dark for few days or even 24 hours is enough. If we keep that leaf for 24 hours darkness, then previously formed starch will be utilized by the plant. So in this way we can D-starch the plant. Place the plant then in the third step, place the plant in a bright sunlight and keep it for the photosynthesis can be performed. Because D-starch is a technique by which we can remove we let the plant to utilize the previously formed starch. But in the second step we have to perform the photosynthesis. That's why we have to keep that plant in the outdoor under the sunlight. Pluck one leaf and mark green and non-green areas on the paper. So suppose there is a you can see there is a leaf is there over here, and this portion are green in color and this portion is reddish in color. That means reddish in color means they lack the chlorophyll, while the greenish in color means they have the chlorophyll. So we have to prove, right now we have to prove the green portion is the only responsible for photosynthesis. Why? Because green portion have chlorophyll, and that's why they only that portion can perform the photosynthesis. Then test for the leaf for the starch. How we can identify that this portion is starch is present, this portion starch is absent? By iodine test. So when iodine react with the starch, it appears as a black to bluish black in appearance. But if there is no starch present, then it will brown in color. It will form a brownish appearance. Observation: the green areas turns blue-black with the iodine. So whenever we put a iodine on the green color, it appears as a blue-black color. That actually conclude us कि photosynthesis is performed, because that's why photosynthesis performed means starch is formed, and starch is formed mean photosynthesis can be done. Similarly, non-green areas mean non-green area no chlorophyll can be found. No chlorophyll means no starch can be formed. If no starch can be formed, how it appears as a blue to black color? That's why it gives a brown appearance. Conclusion. What will be the conclusion? Photosynthesis occur only in green plants. It's all we know that photosynthesis required one thing, that is a chlorophyll, and chlorophyll can be found in only the green portion of the plant. That's why their starch formation occur, and where starch formation occur, they show blue black appearance. And where the chlorophyll is absent, there is no blue to black appearance, because there is no starch formation is there. Chlorophyll is necessary for the photosynthesis. So in this way we can prove that chlorophyll is the vital factor for the photosynthesis. Now come to the next slide. What kind of question you will face in the in your ICSE examination? The first question is, a variegated leaf plant was used to test a requirement for photosynthesis. It was kept in the dark for 24 hours to remove stored starch. What I have discussed, why a 24-hour darkness is very much important, because previously formed starch must be removed. Otherwise, how we can prove that this starch can be formed by the photosynthesis process? Then placed in the sunlight for few hours. Use the diagram to answer the related question. What will be the related question? Now question number 1. Which factor essential for photosynthesis is being investigated using a variegated leaf experiment? Answer, definitely to show the importance of chlorophyll, because if chlorophyll is not there, how photosynthesis can be performed? Now question number 2. What is the purpose of keeping the potted plant in the darkness for 24 hours before exposing it to the sunlight? That I already discussed. If we do not perform this step, this is a very important step. Otherwise, how we can conclude this starch can be formed by the photosynthesis process? That's why previously formed starch must be removed. So that is the importance of these steps. Okay, previously formed starch can be removed. Question number 3. How can the process of photosynthesis be represented using a balanced chemical equation? Now this is the vital question or vital answer student made mistake. What kind of mistake they do in their examination? Like I get a mistake intentionally, that is I do not show any kind of arrow. You have to give a proper arrow. That is, in presence of 6 molecule of carbon dioxide, in presence of 6 molecule of H2O. H2O means water, and CO2 means carbon dioxide. Right? In presence of 6 molecule of carbon dioxide, 6 molecule of water, and along with light energy, especially UV radiation from the sunlight, okay, and chlorophyll. Many students do not write this chlorophyll term and do not use this arrow. As a result, what it will produce? C6H12O6. That is a formula of glucose. C6H12O6. Now you can see that 6 molecule of carbon is there. So 6 molecule of carbon is there. H12 molecule, H2O. So 6 to the 12. So 12H molecule is found. O6. This O6. So in this way you have to balance the equation. Otherwise, anyone if write carbon dioxide plus H2O water, it will react with the sunlight and it will produce C6H12O6, then the equation is not correct. You have to balance it properly. Okay, so the correct equation is 6 molecule of, let me erase, 6 molecule of carbon dioxide along with 6 molecule of water with the light energy, that is the sunlight, and along with the presence of chlorophyll, C6H12O6, that is a glucose formed, and along with 6 molecule of oxygen can be formed. So this is the exact equation, chemical equation of photosynthesis. Okay. In the second question, what would be expected result of a starch test conducted on a leaf from the diagram? It's very easy. Green parts turns blue. Blue to black, sometimes blue or sometimes blue to black, bluish black in color. Why? Because starch, when react with the iodine, it appears as a blue to black. And the portion of the leaf which is not greener in color, that lacks the chlorophyll that does not turn to blue or bluish black. They turns as a brown in color. So by observing the color, the starch, that is called starch stage, we can identify which portion of the plant have chlorophyll and which portion of the plant does not have chlorophyll. Okay, so this is the question number 1. Now second experiment. The first experiment we have discussed about the role of chlorophyll in the photosynthesis. In the second factor, that is the second vital factor, that is the carbon dioxide. How carbon dioxide involved in the photosynthesis process. I have already discussed 3 factors play a vital role. First one is carbon dioxide, second one is sunlight, third one is the chlorophyll. Chlorophyll I have already discussed. Now the second vital factor is the carbon dioxide. What we need to do? Take a plant with a destarched leaf. Same. The purpose of destarched leaf is same. We have to remove, we have to let the plant utilize the entire starch previously formed starch so that we can conduct the experiment carefully. Insert half part of the leaf into the conical flask through a split cork like this way. You can see that half portion of the plants are outside the flask, and half portion of the leaf are inside the flask. Keep the other half of the leaf outside the flask exposed to the atmospheric air. Place a plant in the sunlight for few hours. Now in the flask we need to put a KOH. What is the purpose of KOH? KOH actually absorbs all carbon dioxide present in this flask, because we have to prove the carbon dioxide, the importance of carbon dioxide in the photosynthesis. So whenever we need to prove what is the importance of that, we have to eliminate that factor from that system. Then we can understand the importance of that factor. So similarly, we have to eliminate the carbon dioxide from this flask, and that's why we need to put KOH, because KOH actively absorbs the carbon dioxide. As it actively absorbs carbon dioxide, this entire flask became without carbon dioxide. Okay. After exposure, detach the leaf and test it for the starch using the iodine solution. After that, we need to test iodine solution, because we have to keep it under the sunlight for one day. After 1 day, we will observe that this portion that is exposed outside, they perform photosynthesis, because they can capture the sunlight, they can capture the carbon dioxide from the atmosphere. That's why this portion have photosynthesis. Whereas in the portion that is present inside the flask, they does not have any carbon dioxide. That's why photosynthesis is not performed there. Thank you. In the next day, we will discuss about the very important topic, that is the cell division. Okay, hope you will understand.